Hello, welcome to the BioPharma Finder 3.0 training video. This is Jennifer Sutton, the Software Product Manager. In this video, I'm going to show you how to create a protein sequence for our new top-down workflow. And I'm going to show you an example of trastuzumab, where we bring in the light chain, the FC, the FD, and then we can create proteoforms that will be searched. And please check out some other videos to show you how to use these sequences in the top-down analysis. So here's the home page of the software, and if you click on the Protein Sequence Manager, the tab will open. And in this version, I don't have any sequences in here. And if you do, the table will fill up and show you all kinds of information about the individual sequences. We're going to click on New. Let me scoot this down. Okay, there we go. You should be able to see the screen. Okay, so in the protein sequence editor, what you're going to do here is you can import a protein sequence from a FASTA file, or you can type the sequence or paste the sequence in this area and add it in manually. So let's go ahead and import a protein sequence. And let's start with trastuzumab light chain. So we're going to bring in the sequence. And so you'll see the light chain for the trastuzumab sequence, and you'll notice the category. Now this is new to 3.0, and in 3.0 you need to assign a category. So in this case I'm going to assign it to our top-down workflow. So this helps the software figure out what do you need to do for the different workflows, because the modifications could be different and from peptide mapping, top-down, or intact. So we're going to put top down. And now when we do that, when we actually select the category, the modification tab, site-specific variable modifications for top-down analysis, this page will become active. If you see that this page is not active, then you need to make sure that you select the category that enables this page. So once you're over here, and actually for the light chain, we're not going to add any modifications. We're not going to generate any proteoforms for the light chain, because in the top-down experiments, the light chain has been reduced, and in this example, we don't need to add any modifications. But if you did, you could go through and use this page. And actually, what I'll do is I'll show you how to do that for the FC portion. So let's go ahead and save. And if we cancel, we will see now we have the light chain, and it has the top-down category, tells us the number of proteoforms. It also tells us that there's no modifications, and we didn't assign any glycosylation. So let's do the next one. So we're going to go New, bring this back over. Okay, so now let's bring in the FC. Okay, so this is the FC portion of the heavy chain, and in this one we're going to assign the top-down category. Okay, so if we close this, then we open this back up, now this page is enabled. So the FC region of the heavy chain is the portion of the protein that has the N-glycans. So we can go ahead and assign the N-glycans to this protein sequence. Now something new to the software is we've updated the define modification list. So click on this tab. And when you do this and you scroll through, you're going to see sequences or modifications that are in blue, and those are the default ones that we recommend for peptide mapping. You may also see purple ones. Purple modifications would be ones that you've created in a custom mode. And then if you scroll towards the bottom, something that's new are the glycans. And the glycans are in green. So these are the N-glycans. And what we've done is we've listed all of the N-glycans along with their monosaccharide composition, so you can actually see how they're put together and their chemical formula, and then their average mass and their monoisotopic mass. Now this glycan list is what gets searched in the peptide mapping workflow when you select the CHO or the human database. So you can see there's a large number of different glycans that we actually search in doing that. Now when we've added them as individuals, this enables you to be able to search those or add these as variable modifications for your top-down or your intact workflow. And so we have two checkboxes. We have display variable modifications, and so by enabling this checkbox, they're actually going to show up in the variable modification list. And then you could, if you want, is select default modifications. When you actually check this box, it'll be linked to the default modification button in the variable modification. But for top down, we're not actually going to use this specific column. We're just going to select and enable them here. Now I've gone ahead and chose a few of the glycans. You can see that I have a few of them checked already. So you can hit save. Now, when we go to the site-specific variable modification for top-down, which is kind of a mouthful, but this is where you're going to create your proteoforms. And you can choose the number of modifications per proteoform. In this example, what we're going to do is we're only going to need one, so 1-to-1, because we only are going to look for one glycan. And remember, this is a portion of the FC, so there's not going to be two glycans per FC, it's only one glycan per FC. So here are the list of the variable modifications. Now remember back to this, the defined modification list, that checkbox on the far left. Well, the checkbox on the far left is what you see here. That's what gets controlled in this list. And you'll see here are glycans. Now their filters are in here. You could put in here and you can type N just to simplify this list a little bit, and you can see all the filters that are on the N residue. Now the way this works is when you click one here, okay, you have to check it. The software quickly reads your sequence and comes up with the residue positions of all the N's that could be modified. And so you can select all of them, or if you know, in this case I actually know which one it is. I know it's the 61. I'm going to select 61. When I do that, it automatically goes to our modification list. So now I'm going to say, okay, well, what about this glycan? I'm going to add 61. This glycan, I'm going to add 61. And I can go through the whole list and add 61. Now, if you didn't know the site, so let's say you were looking for an oxidation, and you knew that the protein sequence was oxidized, you just didn't know where the oxidation was, what you could do is you could select oxidation over here, and then you could select all of the methionines or tryptophans and generate proteoforms for all of those. And then you could search all the proteoforms. So what we have here is we have a list, and I've had 10 glycan structures, and I put them all on the 61N. And now what I'm going to do is hit generate proteoform. So what the software does is it goes through and it generates the proteoforms for each one of the modifications that I had in the list. And because this is 1-to-1, that's how it works. And so I should have 10 here. So what I do is I select the ones I want to be searched, and in this example I'm going to search all of them. You don't have to select them all, you can select a small subset of the list. One other thing that's quite nice is when you click on this, it's going to show you where the modification actually is. So in this case it's always on the same position, so it's not going to change. So once I've checked these and I hit save, now it's saved the sequence. When you search this sequence, you're actually going to be searching 11 proteoforms. So it's going to search for all the masses of the different glycans on that residue, and it's also going to search for the unmodified. Let me show you what would happen if we would want to add an oxidation. Let's see what would happen then. So let's do M. So we could click to oxidation, and let's check all these, and let's say 2. And so what it's going to do is it's now added the glycans, and it's also added the tryptophans and the methionines, and we're going to say generate proteoforms. So now if you look, there's quite a long list here, but what the software has done is it's gone through and generated all the different proteoforms based on our configurations. And so in this case here, if we actually click, you can see now that we have a glycan here, and we have a oxidation on methionine 192. So this is a pretty large list. So we have we have over 100 proteoforms. So you wouldn't want to search this really long list. It would be nearly impossible to do this. But if you were looking for, say, a glycan and an oxidation, you could actually filter down this list using the filters that we have here as well. You could say you always want, let's see, I'm trying to think, you could do site 61 because you know that it's always going to have that. And there's different ways that you could filter down the list. But you would choose a subset of this. Anything more than 10 is going to be quite difficult to interpret when you're looking at the results. So kind of as a rule of thumb, you don't really want to search more than 10 proteoforms because you won't be able to actually go through the results in a reasonable time frame to figure out what's correct and what's not correct. Okay, great. So let's cancel this. So now you can see, if you look at, we have the FC and we have our modifications. One thing to notice in the software, if you run into a cell where you have a bunch of dots and you can't see the information, you can hover over this and it'll pop up the modifications. And we have 11 proteoforms. So let's add our last one. And the last one we're going to add is going to be the FT. Again, we're going to import the sequence from the FASTA file. I'm going to select top. Now let me show you what happens if you don't select top down. So see how you can't get into this. Nothing's active until you select a category. So then you go ahead and select your top down category, and again this doesn't have any modifications either. So now we have our protein sequences in the sequence manager, and now we can go to the top down workflow and do our search. Okay, thanks for watching this video, and I hope you found it helpful.